Welcome to Podocyte Talk. In this episode, we explore three pivotal new studies that advance our understanding of kidney disease management, from IgA nephropathy and acute kidney injury to chronic kidney disease with albuminuria. These trials showcase how novel therapeutics, individualized dialysis strategies, and combination therapies can reshape care pathways for patients with kidney disease. We begin with a promising advance in IgA nephropathy, the most prevalent primary glomerular disease globally. This condition, marked by immune complex deposition in the mesangium, is driven by dysregulated B-cell signaling, with BAFF and APRIL, two key cytokines, playing a central role. A recent randomized Phase 3 trial evaluated the effect of Atacicept, a fusion protein that inhibits both BAFF and APRIL, in reducing proteinuria in patients with biopsy-proven IgA nephropathy and persistent proteinuria. In this multicenter, placebo-controlled study, 203 patients were randomized to receive either subcutaneous Atacicept 150 mg weekly or matching placebo. The primary endpoint was the percentage change in 24-hour urinary protein-to-creatinine ratio at 36 weeks. The findings were remarkable. Atacicept led to a 45.7% reduction in proteinuria compared to only 6.8% in the placebo group, a between-group difference of nearly 42 percentage points. This significant result highlights the therapeutic potential of targeting B-cell-mediated pathways in IgA nephropathy. Importantly, the treatment was well tolerated. Although adverse events were slightly more frequent in the Atacicept group, most were mild to moderate, with no major safety concerns. These results offer compelling support for B-cell modulation as a cornerstone strategy in IgA nephropathy, and open the door to a new class of targeted therapies for this challenging condition. Shifting gears to critically ill patients with acute kidney injury, the LIBERATE-D trial explored the impact of a conservative dialysis strategy in individuals who had already started dialysis for acute kidney injury. Conducted across 4 U.S. centers, this multicenter randomized superiority trial enrolled 220 hemodynamically stable patients with dialysis-requiring AKI and baseline eGFR greater than 15 milliliters per minute. Participants were randomized to 2 groups. One received dialysis only when metabolic or clinical indications were present, the conservative arm, while the other continued with a standard thrice-weekly dialysis schedule, the conventional arm. The primary outcome was kidney function recovery at hospital discharge, defined as being alive and dialysis-free for at least 14 consecutive days. In the conservative group, 64% of patients achieved kidney recovery by discharge compared to 50% in the conventional group. Moreover, the conservative approach led to fewer dialysis sessions per week, more dialysis-free days within the first 28 days, and fewer episodes of dialysis-associated hypotension. While some adjusted analyses yielded borderline significance, the clinical signal was consistent. A more individualized, less intensive dialysis strategy may promote kidney recovery, reduce complications, and improve patient-centered outcomes. These findings provide important support for conservative dialysis initiation in appropriate AKI settings and highlight the potential harms of routine dialysis in patients who may still be recovering kidney function. Finally, we turn to the CKD trial, a Phase 2b, double-blind, randomized study investigating the efficacy and safety of combining Balcinrenone, a novel non-steroidal mineralocorticoid receptor antagonist, with the SGLT2 inhibitor Dapagliflozin in patients with chronic kidney disease and albuminuria, conducted across 15 countries. This trial enrolled 324 adults with eGFR between 25 and less than 60 milliliters per minute and UACR between 100 and 5,000 milligrams per gram, all on background renin-angiotensin system blockade. Participants were randomized into 3 arms: Balcinrenone 15 milligrams plus Dapagliflozin 10 milligrams, Balcinrenone 40 milligrams plus Dapagliflozin 10 milligrams, or Dapagliflozin 10 milligrams plus placebo. After 12 weeks of treatment, both Balcinrenone arms showed superior reductions in albuminuria compared to Dapagliflozin alone. Specifically, the 15 milligram combination resulted in a 22.8% reduction in UACR, while the 40 milligram combination achieved a 32.8% reduction. These effects were consistent throughout the treatment period. The trial population was diverse and representative of global CKD demographics. Adverse events were infrequent and comparable across groups, with hyperkalemia reported in 6 to 7% of patients on Balcinrenone plus Dapagliflozin versus 5% in the Dapagliflozin alone arm. Hypotension and renal events were rare, and no serious adverse effects were observed. Notably, both study-related deaths occurred more than 28 days after the last drug dose and were deemed unrelated to treatment. These findings reinforce the complementary mechanisms of MRAS and SGLT2 inhibitors in CKD management. Balcinrenone plus Dapagliflozin not only reduced albuminuria beyond that achieved with Dapagliflozin alone, but also demonstrated a favorable safety profile with minimal hyperkalemia and no unexpected risks. The data support further investigation of this combination in larger long-term outcome trials and highlight the potential of dual-targeted therapy in slowing CKD progression and improving renal outcomes. In summary, these 3 studies, Atacicept in IgA Nephropathy, conservative dialysis in AKI-D, and Balcinrenone plus Dapagliflozin in CKD, demonstrate the expanding frontiers of nephrology, where precision medicine, tailored treatment strategies, and multi-mechanism approaches are improving outcomes across the spectrum of kidney disease. Join us on Podocyte Talk as we continue to bring you the latest breakthroughs in kidney research and care. Stay tuned for episode 34.